This is a quick introduction to the ABO Blood Group and typing. My name is Jean Riddell, and I'm a medical laboratory scientist with my certification as a specialist in blood banking from the American Society for Clinical Pathology. The target audience is MLT or MLS students or anyone who wants to learn about ABO Blood Group. This is a short, basic introduction. The normal blood types in the population are O, A, B, and AB. What about the plus or minus? Why didn't I mention these? These symbols, the plus and then the positive and the negative, which are normal part of when we talk about our blood types, refer to the presence or absence of the D antigen, which is part of the Rh blood group system. So presence or absence of D. So plus means the D antigen is there, and minus means the D antigen is not. ABO blood type inheritance. Simple Mendelian genetics. Codominant expression for the A and B genes. Type O is an amorph that is inherited like an autosomal recessive trait. An amorph is a gene that does not produce a gene product. Type O is expressed when you have two O genes. Type A can be inherited homozygously with two A genes or heterozygously with an A gene and an O gene. Type B is inherited in both homozygous BB or heterozygous BO. So A and B are not dominant over each other, but they are dominant over the O gene. Type AB is inherited heterozygously with one A and one B gene. If you see this little chart, you can see a BO parent and an AO parent, and you can see how the genes cross. You can get AB, you can get AO, BO, and OO from these two types, one person of each type. So if someone is AO or BO, they're just type A or type B. The precursor substance is the starting point for creation of the antigens. This chart demonstrates how the antigens are formed. We start with a precursor substance, and if the person carries, we need to find out if they carry the H gene, the capital H gene. If they carry at least one H gene, they can create H substance. Then we need to know what is their ABO gene, and then which antigens will be presented. If they have an A gene, they will make A and H antigens. If they have a B gene, they will make B and H antigens. And if they have only the O genes, they will only make the H antigens. If they have two lowercase h genes, they cannot make any H substance. Therefore, it doesn't matter what ABO genes they inherit, they will not make any antigens, and they will be made Bombay type. And Bombay type means that you have anti-H and are therefore incompatible with all other blood types. So you could only receive Bombay blood. This chart shows the gene and gene product, the immunodominant sugar, and the antigens produced by the gene. The gene H produces the enzyme alpha-2 fucosyltransferase. The immunodominant sugar is L-fucose, which is added to the precursor substance to create the H antigen. The A gene produces alpha-3 acetylgalactosamyltransferase, and the immunodominant sugar that is attached to the H substance is the N-acetyl D-galactosamine, making the A antigen. The B gene creates alpha-3 galactosyltransferase, which attaches the D-galactose to the H antigen to produce the B antigen. For O, remember, no product, right? They don't make any product, so their immunodominant sugar is just L-fucose from the H antigen, and therefore they only have H antigens. And AB will make both the enzyme from the A gene and the enzyme from the B gene, and therefore they will add both immunodominant sugars to separate H antigens to produce both A and B antigens. In the ABO blood group, if a person makes an antigen, then they do not make the antibody to that same antigen. But if they lack an antigen, they will make an antibody against it. This slide represents the type O blood group. The H antigen is on the red blood cells. The person does not have A or B antigens and will therefore make the anti-A and anti-B antibodies. The immunodominant sugar is L-fucose for people who are type O. This slide represents blood type A. The A antigens are found on the red blood cells, and the anti-B antibodies are found in the plasma. The immunodominant sugar is N-acetyl D-galactosamine. This slide represents blood type B. There are B antigens on the cells and anti-A antibodies in the plasma. The immunodominant sugar is D-galactose. This represents the type AB. Notice that it has two separate antigens, both A and B, not a single AB antigen. Because this person makes both antigens, they do not make any ABO antibodies in their plasma. Let's review this table. Immunodominant sugars. If you're type O, your immunodominant sugar is L-fucose. If you're type A, it's N-acetyl D-galactosamine, and if you're type B, it's D-galactose. If you're AB, it's both the A and the B antigen or sugar. Antibodies for type O are anti-A, anti-B, and anti-A comma B. For type A, it's anti-B. For B, it's anti-A. And for type AB, there are no A or B antibodies. The difference with the type O antibodies, the anti-A and anti-B are both IgM, but the anti-A comma B is an IgG antibody. This is the only ABO antibody that can cross the placenta and can harm the fetus. Although it normally, so if you have a type O mother and say a type A baby, this it's the most common and least severe form of hemolytic disease of the fetus and newborn. So usually the baby is okay. To get blood typing right, let's start with collecting the sample from the correct patient and labeling it properly. The sample must be in the correct collection tube. The sample volume must be sufficient, and the sample cannot be hemolyzed. The reason you can't use a hemolyzed sample is that hemolysis is a positive reaction greater than 4+ in blood bank testing. We centrifuge the sample and separate the plasma from the red cells into a properly labeled tube. Make sure your red cells and plasma samples have the same patient information on them. Follow procedures carefully. Work with only one sample at a time and keep other patient samples in a separate rack from your working rack. Quality control must be completed each day that reagents are used. There should be positive and negative controls. When we are performing blood typing in the laboratory, we perform two tests using reagent antibodies and antigens to mix with patient cells and plasma. Knowing a person's blood type allows transfusions that are significantly safer. If the front type, we will add reagents anti-sera called anti-A, anti-B, and anti-D to 3 individual tubes. Then we will add 1 drop of 3 to 5% solution of the patient's cells. This is then centrifuged and observed for agglutination. The whole button of the cells must be gently shaken off the tube while observing with a light in the magnifying mirror for agglutination. Agglutination is a positive reaction and must be graded 1+ to 4+. For the back type, 2 drops of patient plasma is added to each of 2 tubes. Then A1 cells are added to 1 tube and B cells are added to the second tube. These are centrifuged and observed for agglutination in the magnifying mirror. You should ensure that all the tubes are labeled prior to adding reagents or patient samples. You should read and record the results of each tube individually. Never read them all, then record the results. You should use a pen to record all results in the laboratory. You should never scribble to make a correction. All corrections should have a single line through them, an initial and date. You should be able to see what the correction is, no pencils or erasing. This is an example of a front type on a card. The left side is anti-A with the patient red cells, and the right side is anti-B with the patient red cells. Anti-A is blue and anti-B is yellow. The patient cells are diluted with blood bank saline. This example is from a whole blood finger stick. Notice when anti-A clumps, then A is part of the blood type, and when anti-B clumps, B is part of the blood type. If there are no clumps, the person is type O. The back type is the patient's plasma mixed with reagent red blood cells. We use A1 cells and B cells. If the patient reacts with A1 cells, then A is not part of the blood group. If the plasma reacts with B cells, then B is not part of the blood group. It will look reverse from the front type. Notice the forward and reverse typing are reverse reciprocal. If they are not, you must check your labels and repeat testing. Let's go through the chart. We can see the forward typing, you're adding patient cells to reagent anti-A and reagent anti-B, and reverse typing, you're adding patient plasma to reagent A cells and reagent B cells. So type AB would react with anti-A and anti-B, but the plasma from that person would not react with A cells or B cells. Type B will not react with anti-A, but will react with anti-B. And type B plasma will react with A cells, but not with B cells. Type A will react with anti-A and not with anti-B. And A plasma will react with B cells, but not A cells. Type O will not react with anti-A or anti-B, but will react with A cells and B cells. Summary: ABO blood typing. There are 4 ABO types: O, A, B, and AB. Types are defined by immunodominant sugars. Gene products are enzymes. Accuracy is extremely important in testing. Red blood cells have antigens on them, and plasma has antibodies in it. Forward and reverse typings must correlate. If you have any questions, please leave them in the comments below. Remember to like and subscribe and share with other students. Thank you for attention. We use the Harmoning Modern Blood Banking and Transfusion Practices as our reference.